Good morning, everyone, and welcome to today's MPG Primer session. It's my pleasure to introduce our speaker today, Dr. Victor Nauffal, MD, MSc, who is a cardiovascular medicine fellow at the Brigham and Women's Hospital and a T32 postdoctoral fellow in the Ellinor lab at the Broad Institute of Harvard and MIT. His research focuses on examining the genetic basis of cardiac arrhythmias, utilizing population-based genetic studies. He has a particular interest in atrial fibrillation and sudden cardiac death. He graduated from medical school at the American University of Beirut in 2013 and completed a Master of Science in Genetic Epidemiology and Statistical Genetics at the Harvard T.H. Chan School of Public Health in 2021. He moved to the United States in 2013 to pursue a 2-year postdoctoral fellowship in the Johns Hopkins Murex Postdoctoral Fellow program, where he focused on enhancing risk stratification for sudden cardiac death in patients with heart failure. He completed his internal medicine residency training at the Johns Hopkins Osler House Staff Training Program before moving to Boston for cardiovascular medicine training. In his exciting next step, he will embark on a cardiac electrophysiology subspecialty training at the Brigham and Women's Hospital starting July 2022, and we look forward to his ongoing work in the field of cardiac arrhythmias. We welcome him today for his talk, Polygenic Risk in the Context of Monogenic Diseases, and he's happy to take your questions throughout the talk. Thank you, Victor, for joining us today.
Good morning, everyone, and thank you, Sarah, for the kind introduction and for the invitation to take part in this series. I'm really excited about this and hope the audience finds this session to be informative. So as you mentioned, I'll be talking today about polygenic risk in the context of monogenic diseases. And as Sarah just mentioned, I'm really happy to take any questions, or please feel free to interject with any questions throughout the talk. And hopefully there should be ample time at the end to go over any questions and have a thorough discussion. Just as a disclaimer, I am a cardiologist, so many of the examples that I will give throughout the presentation to illustrate concepts will be from the cardiovascular field. I will try my best to use non-technical terms for the non-clinicians, but again, feel free to interrupt with any questions. I have no disclosures, and the outline of the presentation will be as follows. I'll first sort of go over background of the genetic architecture of common and rare diseases as we understand them today, introduce the concepts of penetrance and variable expressivity in rare monogenic diseases, and then finally discuss the role of polygenic risk in modulating disease susceptibility in rare monogenic diseases. So first of all, starting with the genetic architecture of common and rare diseases. So when we think about disease or determinants of disease in the population, there's two broad buckets of factors that we usually consider. Environmental factors, which usually are exogenous factors that encompass anything from socioeconomic status to social determinants of health, some epidemiologic risk factors, and other comorbidities, lifestyle, diet that potentially contribute to disease. And genetic factors thought to be more of endogenous factors that could also contribute to disease. And these factors don't act in isolation. There's quite a bit of sort of interplay and interdependence between these two factors as they contribute to disease expression in the population. And for example, environmental factors through epigenetic modification can alter the association of the genetic code with disease in the population, and the reverse is true as well. But for the rest of this talk, I'll focus mostly on genetic factors and their contribution to disease in the population. And we usually think of two broad sort of buckets in terms of genetic contribution to disease in the population, the contribution of rare variants and common variants. And in a simple sort of construct for disease, we usually think of rare variants as contributing to rare monogenic disorders with a low prevalence or rare prevalence in the population, defined as less than 1 in 1,500 or less than 1 in 2,000, depends on where you get the definition from. The genetics of those disorders are usually thought to be simple, Mendelian genetics, and these disorders are usually unifactorial in etiology. And good examples are the long QT syndrome or phenylketonuria, and there's a slew of other examples as well. And we think of common variants as predominantly contributing to common complex diseases, which have underlying complex genetics and are multifactorial in etiology with a number of biologic pathways sort of contributing to disease expression in the population. And examples of those disorders are hypercholesterolemia and coronary artery disease. However, this simple construct does not necessarily fully capture the genetic contribution to disease in the population. And we've appreciated over time that there are rare variants that contribute to common disease, such as monogenic forms of hypercholesterolemia, such as familial hypercholesterolemia. And then more recently, we've come to appreciate the contribution of common variation to rare monogenic disorders. And that will be sort of more of the emphasis of this talk, and we'll elaborate more on this concept. And again, rare variants and common variants don't act in isolation, and there's quite a bit of interplay between rare and common variation and as they contribute to disease. And just as to highlight, when I talk about variants here, I don't necessarily only mean single nucleotide polymorphisms, but these could also be structural variants or copy number variants as well. I'm going to take a moment here just to sort of go over this concept of the inverse association between allele frequency in the population and effect size. And this is due to the action of negative selection, which usually leads to deleterious alleles that have large effect size that are usually sufficient to cause disease in the population or under the action of negative selection, and over generations are filtered out from the population. And this is why we see that alleles with large effect size that are usually sufficient to cause rare Mendelian diseases have a rare allele frequency or low allele frequency in the population, whereas alleles with small effect size usually have higher allele frequency or uncommon in the population and tend to contribute to common diseases. However, this should not be understood as that common variants only contribute to common disease and rare variants only contribute to rare disease, because it is also true that rare variants contribute to common disease and that common variants contribute to rare diseases. And in this overall framework of the inverse association of allele frequency with effect size, there are a few exceptions. It is sort of a good broad rule to follow that is truly followed, but there are a few exceptions to that. And there are examples of common variants that have high effect sizes that are common in the population. These are variants that are usually under the sort of the pressure or the force of positive selection. And one example of those are common variants that contribute, for example, to sickle cell trait in African ancestry populations, which continue to have a high effect size and are common in the population. That's because they promote a survival advantage in those populations. And so I wanted to just sort of go over a construct for common diseases or a common disease model. And in sort of the field of population genetics, it's hard to generalize, but I think this is a sort of a good construct to think about common diseases. And I put this schematic together. Again, doesn't necessarily encapsulate all common diseases, but it's a good framework to think about common diseases and the contribution to common disease. So there are both genetic and environmental factors that contribute to common disease. And there's a small minority of cases of individuals who have monogenic forms of common diseases, but the majority of patients either have a polygenic predisposition to common disease or environmental factors that predispose them to develop common diseases. And a good example of a common disease, just to put things in context, is hypercholesterolemia. So it's a disorder of cholesterol metabolism associated with elevated plasma cholesterol levels and increased risk of atherosclerotic cardiovascular disease. And that usually means, in sort of lay terms, increased risk of heart attacks or strokes and other disorders. Again, it's a common disorder, so it's a reported prevalence of around 1 in 12 in the population. And when you look at the flowchart on the right, which is a study from the Mayo Clinic Biobank that tried to look at the etiologies of primary hypercholesterolemia, you find that a tiny fraction of cases have a monogenic architecture, only 1.5%. And a sizable proportion have actually a polygenic etiology, around 15.6%. And then the majority of cases, around 83%, don't have a yet identified genetic etiology. And those individuals could perhaps have significant environmental risk factors or environmental exposures that predispose them to disease, or could have some genetic factors that we have just not yet identified. But this is overall sort of the construct of common diseases, with a small contribution from monogenic monogenic etiology, a larger proportion of polygenic predisposition, and sort of a significant contribution of either environmental exposures or some genetic factors that we haven't discovered to date. On the other hand, this is a sort of moving on to the rare monogenic disease model. And so in this dotted line, I show the distribution of affected individuals. You can see that the peak is lower than the common disease model because this is a rare disorder, there's less affected individuals. And again, there's both environmental and genetic factors, but the majority of individuals have a monogenic architecture of disease. However, there are a number of individuals who have polygenic architecture or polygenic predisposition to developing rare disorders. And we were attending and more recently to understand that more clearly. Again, an example of a rare monogenic disorder is long QT syndrome. And so it's a disorder of cardiac repolarization that's associated with prolongation of the QT interval. Just to explain what that is, the QT interval is an interval that is usually captured on an electrocardiogram, which is a heart tracing that measures the electrical activation or conduction through the heart. And it is an interval during which the heart resets and sort of gets ready for the next heartbeat. And the longer that interval is, the higher someone's predisposition to develop lethal heart rhythms and to suffer sudden cardiac death. Again, this is a rare disorder. Its prevalence is around 1 in 2,500 in the population. And as a rare monogenic disorder, predominantly 80% of the cases are secondary to have a monogenic architecture that are secondary to rare disease-causing variants, usually in 3 ion channel genes, KCNQ1, KCNH2, and SCN5A. And whereas for example, in common diseases such as hypercholesterolemia, we're able to sort of quantify the polygenic contribution more robustly to those disorders, we still don't have strong polygenic instruments for rare diseases. However, more recently, we have started to perform genome-wide association studies of rare disorders and have identified for long QT syndrome, for example, that the common variant SNP heritability is around 15%, which is that suggests that there is a common genetic or polygenic sort of contribution to this rare disorder. So in summary so far, both genetic and environmental factors in varying proportions contribute to disease, and I think that's established. And the contribution of genes versus environment varies from one population to another, and that's a topic for a different discussion. But for the most part, both the genes and environments interact and contribute to disease in the population. And when we're thinking of sort of rare diseases and common diseases and their genetic determinants, it's important to understand and realize that both rare and common variants contribute to both forms of disease, despite to a varying degree. And so far, I think we've been successful sort of in quantifying the contribution of common genetic or examining the contribution of common genetic variation to common diseases through genome-wide association studies. But we still haven't been able to do that as thoroughly for rare diseases, mainly because of limitations of power and sample size that are sort of imposed by the rare nature of those diseases. And secondly, I think for a long time, understanding or acknowledging that these were monogenic diseases, there was less interest in sort of exploring the polygenic architecture of those disorders. So now I'm going to move on to talk and introduce concepts of penetrance and variable expressivity in rare monogenic diseases. So penetrance by definition is the proportion of individuals with a particular genotype who express the associated phenotype. And so penetrance could be a number from 0 to 1. So you could have a variant, for example, that is expressed that leads to disease in every individual who carries that variant, and that would be a completely penetrant variant. But most of the time we have a phenomenon of incomplete penetrance in sort of rare monogenic disorders. There's a proportion of individuals who express disease and a proportion of carriers of that variant or risk allele that do not express disease, and that's now understood as incomplete penetrance. And the mechanisms behind incomplete penetrance are really poorly understood to date. The other concept is the concept of variable expressivity, which is the heterogeneity in disease manifestation or the severity of the disease among affected individuals with the same genotype. And I think I really introduced these two concepts as challenges in our understanding in our clinical management of patients with rare monogenic disorders. And I'll sort of later on touch on ways that polygenic risk may sort of enhance our understanding of those two phenomena. But just to put this into context, I thought I'd give a clinical example of sort of incomplete penetrance. Someone who I saw in clinic a couple months ago was a 37-year-old male who presented for evaluation of long QT syndrome after he was incidentally found to have this missense variant, missense rare pathogenic variant in KCNH2 that's been associated with long QT syndrome. So this individual came about knowing that he was a carrier of this variant after his 7-year-old son, who underwent whole exome sequencing for a non-cardiac indication for a musculoskeletal disorder, was found to be a carrier of that variant. And that prompted cascade screening in first-degree family members and identified the father as a carrier of this variant. When I sat down with the patient, he had no history of any cardiac arrhythmias and no history of resuscitated cardiac arrest. And sort of reviewing the family history over 3 generations, there was no evidence of sudden cardiac death or any sort of phenotypic manifestations of long QT syndrome. But since this is a pathogenic variant, it required a little bit more sort of clinical investigation in this case. And I don't want to sort of bore you with the details of clinical cardiology, but this is a sort of more thorough evaluation of the father and the son. And you can see that in those heart tracings that the QT interval, sort of a hallmark of long QT syndrome, is both normal in both individuals. Usually anything less than 440 is considered to be a normal QTc interval. And so those intervals were normal in both individuals, and both are carriers of this pathogenic variant. We did a more sort of thorough investigation where we measured the QT during exercise and through recovery for both the father and the son, as you can see here in green and red, respectively, for the father and the son, that the QT interval during exercise and during recovery remained within the bounds of normal limits, suggesting that those 2 individuals who carry a pathogenic variant had no sort of evidence of sort of clinical signs of the disease, and on further evaluation had no evidence of long QT syndrome. And this is sort of in contrast to the family where this variant was first described and has been described to be pathogenic in different sort of case series as well. You can see from this pedigree where infected individuals are shaded in black that the variant co-segregates with affected individuals, and that this variant is highly penetrant, with most individuals who carry this variant expressing disease. Maybe one exception to that is individual number 13, who carries the variant and does not develop the disease. So hopefully this illustrates the sort of the challenge of incomplete penetrance, where the same variant in one family over 3 generations has no evidence of clinical long QT syndrome, and that same variant is pretty highly penetrant in another family. And there are certainly environmental factors that could modulate penetrance. Really, we have been unsuccessful in sort of pinning down these environmental factors so far. And potentially there might be some polygenic architecture that modulates that we'll talk about as we go through this presentation. And this is a clinical challenge because this individual who's a carrier of a pathogenic variant will have to come back for repeated clinical evaluations to understand when and if they develop penetrant or manifest disease. So it'd be interesting to understand what causes penetrance and what sort of impedes penetrance. So hopefully, as I sort of illustrated some of the problems in sort of approaching and managing monogenic disease, next I'll move on to talk about how polygenic risk may have sort of enhanced our understanding of rare monogenic diseases. So how can common variation contribute to our understanding of rare monogenic diseases? So first of all, I think one area where polygenic risk may be helpful is that there is a sizable proportion of cases that have no yet identified rare variants associated with disease. So we clinically sort of refer to them as genotype negative. I don't love that term. These are individuals who have a manifest disease, for example, long QT syndrome, who, if we perform gene panel testing today, evaluate for the genes that we know are associated with long QT syndrome, we don't identify any pathogenic variants associated with long QT syndrome. And this could be that there's individuals that there are elusive rare variants associated with long QT syndrome that we just haven't discovered yet, or that there might be a more complex polygenic inheritance pattern in those genotype negative cases. And to sort of give you some numbers and sort of how many patients actually are genotype negative so far. So for example, long QT syndrome numbers as high as 20% have been reported for individuals with long QT syndrome who don't carry rare variants that we've identified so far. And for another monogenic disorder, hypertrophic cardiomyopathy, slightly more common than long QT syndrome, this is around 60% of cases where we don't identify rare variants associated with hypertrophic cardiomyopathy in the clinic in cases who have evidence of this disease. So that's one area where polygenic risk may be helpful. And the other area, as I just mentioned, is understanding the mechanisms that mediate incomplete penetrance and variable expressivity among monogenic rare variant carriers in the population. And so one sort of appeal of polygenic risk is that it sort of integrates multiple pathways related to disease and sort of could allow us to understand someone's susceptibility to develop disease once they have a rare variant associated with a disease, and that could modulate penetrance and also could modulate the severity of disease among carriers of rare variants. And so now I'll move on to talk about some of the work that's sort of really recent and happened over the past year or two that sort of elucidates some of the common genetic contribution to rare monogenic disorders. And I'll start with the long QT syndrome as a case example. So this is a GWAS that was published around a year and a half. It's a case-controlled GWAS of around 1,700 cases and 10,000 controls of long QT syndrome. And just want to sort of highlight that if you compare this to some of the common disease GWASs, for example, for atrial fibrillation in 2018 or for type 2 diabetes in 2018, you're talking there about something around 65,000 cases and 440,000 controls. And just to highlight the limitation that rare diseases impose on us in terms of the power of the GWASs that we're able to perform. And that's reflected in the findings. And so in this GWAS of long QT syndrome, there's only 3 genome-wide significant SNPs that were identified, and then 1 subthreshold SNP that was identified. However, we did find that the SNP heritability from this first long QT syndrome GWAS was around 15%. So really highlighting that there is a sizable and a reasonable common genetic contribution to this rare disorder, what we think of as a monogenic disorder. However, the reduced power may sort of hinder our ability to generate polygenic instruments that are strong enough to sort of better characterize polygenic contribution to disease. However, until we have, and that could be a limitation until we have larger sort of genome-wide association studies of rare diseases where we have sort of enhanced number of cases to sort of detect more signal. But until then, one potential option is to leverage endophenotypes that are closely related to rare disorders as a window to understand the polygenic architecture of a rare disorder. And so I think we have successfully and nicely done that for long QT syndrome. And I think long QT syndrome is unique in the fact that the QT interval is sort of central to making the diagnosis and highly correlated with the phenotype itself, with the disease itself. And so if you compare, for example, the loci from the QTC GWAS to those in the long QT syndrome, you find that the 3 genome-wide significant loci from the long QT syndrome are all genome-wide significant for the QTC interval. And interestingly, the top locus for both the QTC and the long QT syndrome has exactly the same SNP. And if you look at the common variant genetic correlation between long QT syndrome and the QTC, it is reasonably high at 0.4. And if you look at this plot, there's sort of strong correlation between common variant effect sizes for long QT syndrome and the QT interval. So that possibly highlights a sort of smart approach to use endophenotypes to sort of understand the polygenic architecture of a rare disease. And this is what LaRouche and colleagues have done in that initial seminal paper where they published the first GWAS of the long QT syndrome. They used a polygenic risk score for the QT interval and found that by doing so, they could actually sort of identify increasing susceptibility to long QT syndrome across increasing quartiles of the polygenic risk score for the QT interval, which is pretty interesting and sort of a interesting way to support the sort of polygenic architecture of long QT syndrome or contribution to long QT syndrome.
We have a question that's just been posted, Victor, exactly pertaining to the question of genetic architecture, and I will read it now. The question is, how much is known about a combinatorial effect of a small number of rare or common variants as opposed to a full polygenic contribution in the context of rare disease?
Dan, can you just go over the question? Sarah, can you go over the question again? Sorry.
Of course, yes. Let me try to voice it more clearly. Sorry. So the question is, how much is known about these 2 different possibilities? The first being a combinatorial effect of a small number of rare or common variants, and the other, a possibility being a full polygenetic contribution in the context of rare disease.
Yeah, and I think that's a great question. I think the answer to that, I think we don't fully understand that, whether there's just a pure polygenic form of disease or it's a polygenic form interacting with rare variants that lead to disease in sort of rare disorders. think if there's one way to sort of approach this. If we know these are rare disorders, so the polygenic contribution could not be very sizable, otherwise those diseases won't be that common. Well, it won't be rare, it will be more common. So I think it's a combination of both. There is a suggestion of a potentially just polygenic form, especially in those who don't have rare variants. But we can't completely say that that's truly the case, that it's only a polygenic architecture, because we don't know if there's rare variants that are not just yet discovered. So I think it's a combination of both. I think there's significant interest in sort of the combined effect of polygenic risk and rare variants sort of contributing to disease, but there's also the possibility of just a pure polygenic deterrent component, albeit that would not be a major component, otherwise those would not be rare diseases.
Thank you very much.
Okay, all right. And so, as I just said, highlighting here that this polygenic risk score for the QT interval was able to risk stratify, or at least to identify increasing susceptibility to disease across increasing quartiles of the polygenic risk score. And they took this a step further and sort of looked at the distribution of the polygenic risk score across controls, genotype positive cases, and then genotype negative cases. And that might sort of come to answer the question that was just posed, that sort of the higher polygenic risk score among genotype negative as compared to genotype positive cases potentially could suggest that there is maybe a sort of a predominant polygenic component to long QT syndrome. Again, that we cannot be 100% certain because we don't know that they're truly genotype negative, but that is the sort of the higher polygenic risk score in that subgroup may sort of suggest that maybe there is truly just a polygenic form of long QT syndrome. And the other interesting point is that the higher polygenic risk score among genotype positive cases may somehow allow us to extrapolate the role of polygenic risk in potentially mediating penetrance in long QT syndrome, which would explain why there's a higher polygenic risk in those genotype positive cases as well. Our group tried to sort of address the polygenic and monogenic contribution. Obviously, these studies I just mentioned are case control studies of long QT syndrome. We tried to sort of look in the general population among sort of all comers that are potentially carriers of monogenic rare variants. The only difference that we didn't have is that we did not have sort of ascertainment of long QT syndrome in this population, but we used the QT interval as a proxy. And what we did is we performed a large GWAS in the UK Biobank and transferred that GWAS to TopMed, which is a cohort where we had whole genome sequencing data. And what we did is we characterized rare variants in long QT syndrome genes and then scored the TopMed cohort using the QTC polygenic risk score and looked at the contribution of the polygenic risk score and monogenic rare variants to the QT interval in that population. And what we found that there was both a monogenic contribution and a polygenic contribution to the QT interval. And interestingly, what we found is that among monogenic rare variant carriers, that the extent of QTC prolongation was double among those who had high polygenic risk. Again, we can't make any statements about penetrance here because we only are looking at the QTC, but given that it's highly correlated with the long QT syndrome, one possibility is that polygenic risk could modify sort of either disease severity or penetrance among rare variant carriers in genes associated with long QT syndrome. So just to summarize some of sort of the points that I went over, I think I hope I had stressed that common variation does contribute to monogenic rare diseases, and that the evaluation of highly genetically correlated endophenotypes may be a window into exploring the common genetic architecture of rare monogenic diseases. The additive contribution of common genetic variation in the form of polygenic risk scores contributes to monogenic rare disease susceptibility, especially among non-carriers of rare disease causing monogenic variants, as I just showed in that long QT syndrome paper. And the higher polygenic risk scores among rare variant carrier cases may suggest that polygenic risk score contributes to penetrance of monogenic rare variants. And then I wanted to talk a little bit about another sort of example of a monogenic disorder, hypertrophic cardiomyopathy, where sort of over the past year, there's this GWAS of hypertrophic cardiomyopathy that again sort of showed the contribution or demonstrated the contribution of common genetic variation to hypertrophic cardiomyopathy. They identified 6 loci associated with hypertrophic cardiomyopathy. And interestingly, they sort of also demonstrate in this study potentially a role for the polygenic risk in sort of modulating or contributing to variable expressivity. So what they did is they looked at individuals with HCM who were carriers of rare variants associated in genes, in sarcomeric genes or genes associated with HCM, and found that the polygenic risk score was able to stratify the lifetime risk of events among individuals who were carriers and had hypertrophic cardiomyopathy, suggesting that there is a role for polygenic risk potentially in sort of defining variable expressivity or sort of elucidating the contribution of the polygenic risk score to variable expressivity in monogenic rare diseases. So in conclusion, I just hope I stressed that both common and rare genetic variation contribute to disease, despite at varying proportions depending on the type of disease that's being studied, and that there is a polygenic architecture of monogenic rare diseases that has been more recently elucidated, whether it is a unique pure form of those rare diseases or it's an interaction between polygenic risk and rare variation. I think it's a combination of both, but sort of more studies are needed to fully shed light on this phenomenon, and that polygenic risk may contribute to rare disease susceptibility. Especially sort of interest is among non-carriers of monogenic rare variants, and then finally that polygenic risk among carriers of rare variants may contribute to penetrance as well as variable expressivity of rare diseases. And I'm happy to sort of talk more or take any questions.
Thanks so much, Victor, for a really amazing talk. A question just was posted, and I will read that now, which is, first of all, awesome talk. And then secondly, can you comment on how common the finding that monogenic and polygenic contributions are strictly additive across phenotypes?
Yes, I think that is a great question. I think for the most part, some of the work that we've done in some work, I mean, it's been published in sort of other groups, I think within the Broad, Akil Fahad has done some work, and Ahmed Kara's group as well, that show that there is that polygenic risk and monogenic risk, there is sort of an additive effect that contribute to disease. Most of the time that seems to be true. And so I think that is true. I think the bigger question is there a multiplicative effect? And is there sort of effect modification occurring between the sort of polygenic risk and monogenic risk? And I will say that is not always true. Sort of in some of the work that we've done, we saw that was true in some cases for the QT intervals and not in other cases. And there might be some specific, so I'll give you sort of an example from the work that we've done, is that depending on sort of pathways or sort of components of the polygenic risk score that we tested, we found effect modification, whereas for others there was not. So I think there might be biologic pathways that the polygenic risk score captures that may sort of interact in a multiplicative fashion with monogenic risk. But overall, there seems to be a consistent additive effect, and maybe if we isolate specific pathways, there might be sort of a sort of interaction on the multiplicative scale. But I do think that the additive effect has been consistently shown, at least in most of the studies that I've reviewed.
Thank you so much. And then finally, just one sort of more a forward-looking question in terms of what your thoughts may be on polygenic risk scores in sort of long-term understanding of rare disease and how we can go about in the future collecting relevant data and inferring environmental and genetic, be they mono or polygenic contributions to rare disease.
Yeah, I think so. I think as we discover more, I think we might have issue with calling these disorders just monogenic. I mean, this is particularly true for hypertrophic cardiomyopathy, for example. There's a significant portion of patients so far that we don't find rare variants associated with disease in. So I think as we go forward and learn more about the polygenic architecture of those rare diseases, I think we sort of might sort of change the framework by which we look at those diseases, not just understand them as monogenic diseases, but sort of understand that there's a polygenic contribution. I think sort of moving forward, I think there is really a need to sort of join forces and sort of enhance the size of our GWASs to understand the contribution of common variants to rare disease. I think that would be very informative into sort of elucidating the full architecture of the genetic determinants of those disorders, and also sort of understanding, sort of hopefully sort of bridging some of the gaps in our current understanding of rare diseases, especially in terms of penetrance and variable expressivity.
Thank you so much. That the thought about rethinking the mono part of monogenetic disease seems like a really important one to keep in mind. So thank you so much, Victor, and thanks to everyone who attended and raised questions, and we will see you at a future MPG Primer. Thank you.
Thank you.